Sydney would chair, and so originally it was stated that the University of Sydney would chair and UCL would take minutes. I don't mind chairing. Dimitri, would you take the minutes then?
Yeah, that was the intention. I would take minutes anyway.
Okay. So then, well, if Matt is still on holiday and from the University of Sydney, Anita is the only one, then, oh, Hang is there too.
Hi, Hank.
So then, Hang, you could chair?
Yes, but do let me have a quick read because I have actually been up to date. Been very busy with my thesis, but give me a moment. All right, hi everyone. I'm just gonna go through the agenda. So. Okay, so first one is, so I have completed the selection for epichem molecules. So the next step is actually to send this to Wendy, which is the suggestion of compounds for to be asked from the epichem library. So does that mean that Wendy, you are going to be sending the request to epichem?
No, I think this meant that Matt was going to ask them from epichem and to get the molecules to me for screening.
Okay, so that's actually to Matt. I was gonna. Do I need to do anything to? Because I'm not sure. We've already got the selection of compounds. So will Matt be doing that, or do we actually need to do something before that?
I suggest we put this as an objective for the next meeting so that he would, like, when he's back, he knows that he needs to contact them. That would be probably the easiest way.
Yep. Okay.
Yeah, because anyway, these will not be for this season, larvae season anymore. So they will be screened in vitro, but probably not in the larvae. So yeah, and the current manuscript will not wait, I think, for this edition.
No, I think we've already have a cutoff, and these will be going in there because otherwise it's gonna be dragging on for a few more years. Let's just get it out of the way. I am working on it.
Yes, I looked at it yesterday actually. So I added all the references because they went. There was something which went wrong during the copy pasting. So I did add the PubMed ID.
Yeah, I think that works quite well. I actually, since you mentioned it, so I just realized I also cannot update my my reference list inside Dropbox because I used to be able to do it outside. So it might be a Dropbox compatibility issues. I'm happy to compile the final reference list. If you know, is it like how you've already got all of the PubMed ID? I can just use that to grab the actual paper. I think as long as I have that, I should be okay for me to compile the final list. I'm happy to do that. It's just that because I don't know, otherwise I won't have the papers to the list of papers actually compile it otherwise.
No, that's why I did the PubMed ID, because usually everyone can access that, and that's easier. I also made, maybe it is a little bit ahead of the agenda, but I made the Excel file, which should be accompanied as the supplementary material for this manuscript. It's still the same number of compounds, but I noted that we lacked a few log-D values. So Louisa already created those. So I will upload that later today and rerun the in vitro analysis to get to the final figure. And that's all the actions I think still needed from Ma and me.
Okay, for the manuscript. Okay, yeah, sounds good. Actually, since we're on that, we'll just talk about that to get it out of the way as well. So I'm still working on the manuscript. Actually, haven't finished with it yet. Hopefully, I'll get it done by tomorrow, Thursday. I just need to go through and clean all the comments. There's a few left that just require me to finish. And I think Matt also make a few mistake of assigning some of the comments to me. It's actually to Dimitri. So I think they
vice
should
versa as well.
Yeah, it's actually quite funny. So Dimitri, if you want to work on those as well, because I don't think they will clash with me. So I think we can work on both of those at the same time.
No problem, because we have agreed with Matt as absolute deadline for this. So by the time he comes back from holiday, it should be cleared off, because he needs to review it like in full. So that's
okay. Yeah. So whichever one is supposed to go to you, Dimitri, I've already reassigned that to you.
It's the same file. It's synchronized in Dropbox, so it's updated.
Yeah. So I just put a comment, but sometimes Matt was telling me to fix something on your compound, and I can't do it. So
comments backwards as well.
Yeah. So I think we can both work on that. I'll should be finishing this latest Thursday this week. So just to get that out of the way.
Personally, I have mainly NMR issues left to resolve, so that's checking on the existing NMR comments mainly, because I did like most of the corrections like in July.
Yeah, so I created yesterday a version dated from yesterday when I answered all the comments, because it asked me to create a new name. But I think I was the only one working there yesterday, so in Dropbox.
Yeah.
I saw your updates on the go, like while I was looking at the manuscript. I saw how you updated the.
Oh, okay.
Yeah.
Do we know who currently put the reference list at the end? Because I think it's slightly out of sync. I will manually do one at the end, but I'm just making sure that I might not be deleting anything that I'm supposed to.
I think the current reference list was the endnote created by me, but it doesn't work in Dropbox.
Yeah.
So that's why I made a comment in front that everywhere where I put the PubMed ID, you can remove the reference, but I couldn't remove it in Dropbox. It wouldn't allow me.
Okay, so I think when I'll do the reference list at the end, I will ignore that and I'll create one for everyone. I think that would work a bit better.
Yeah.
Okay.
Just a quick question, Wendy, about the Excel file that you mentioned yesterday, that you asked me and how to have a look at. I'm slightly confused. It has many more molecules than we have in the manuscript. So what was that file for?
That well, the file I shared has a few more molecules, but I removed all the duplicates. So, for instance, for MycetOS 001, we have 3 versions of that molecule. So 1, 2, and 3. So for the analysis, where we correlate log D to the in vivo activity or the in vitro activity, I only use the first one. So 0,1, because otherwise you get 3 entries of the same molecule, and that confuses the statistics. For
but
now,
the number of entries is 197 at the moment, and that's way above like what we have in the manuscript. So there's no way we have so many molecules. That's what's confusing to me personally.
197, yeah, we do have 185 in the manuscript.
Really?
And if you remove the duplicates, then you are at 185, because I double checked several times yesterday. But for now, because that was still an open question why I wanted you and Hang to check, I decided to leave those numbers in the table. We can also choose to remove the duplicates of each compound, because of course MycetOS 001 is 3 times the same molecule. but made 3 times and evaluated 3 times. But for the analyzers, it's only considered once, of course.
For much quicker response from everyone, I have a quick suggestion. Would it be easier to upload this Excel file to Dropbox, and then every of different parts, different members of the manuscript can go into that file and update it on the go as well? So say, for instance, like highlight the line with the entry which is actually in the manuscript, so that we know which one is in and which one is not going into that list. Would that be easier?
That's the strange thing, because I am not a chemist, so I cannot see which actually is displayed as a molecule which is made. But for the analyzers, linking the in vitro properties with the in vitro susceptibility and the in vitro and the in vivo efficacy, I analyzed all of them. So the 185 compounds we screened, or 180 compounds we screened, I cross-refed their log D, for instance, with their in vitro activity. So they were all evaluated. So they are all there, but of course they are not all synthetically made. So some of them are from Epichem, and others are from Hang, and others are from you. And I'm not a chemist, so I cannot see which molecule is in the method section. I can see only by code, but I left that up to actually Matt to determine if we need to, because as far as I'm concerned, all 185 needs to be in that table.
Yeah. In that, if just a quick note on that. So whatever is in the experimental section, those are the ones that are made by Dimitri and myself. If it's not synthesized by us, it won't be in there, because we wouldn't have all of the details and all of the methods for it. So those specific ones are from Dimitri and myself.
Yeah, so maybe because now it says made by, and then we should say Epichem for the details there and refer to that manuscript.
Yeah, so it should be similar to what we have on the master file.
Yeah,
where
so I copied the master list and removed all the entries from the other series.
So Wendy, just a quick response to what you're saying. So for instance, myas 009, so I find it like in your list, and then we do something like this highlighting. Would that be a good version? And then this file is synchronized in Dropbox, so whenever like you or anyone else opens it, like people can see, and then we can match which molecules are in the manuscript, like with this list. Would that be helpful?
I guess so, but here you can also see what I meant with some molecules we have multiple versions. So myas 001, you see 0102 at the end.
But then we would highlight which one has been tested and which one we mean in the experimental section from this list.
But they are all tested.
They've all been tested. That's the thing.
Yeah, I get your point.
So the original reason that some of these are duplicated is some of them are resynthesis. So they just mean that, for example, I or Dimitri need to check the synthetic route. So actually remake some of these hits, and some of them are also just recheck that, like recheck the efficacy as well. So that's why some of those are duplicated. But yeah, you should be able to tell from the master list where these compounds belong. So I don't think that should be a problem. We just need to tell who's it's from. So either UCL or Epichem. I think that should work. I don't know. Maybe just double check with Matt.
Okay.
Yeah.
All right. Let's go back to the beginning. So the next point is to evaluate the latest models of this, but there is some delay or there's some problem. Is that do you have an update for that?
We don't have the update of in vitro data, but I have update of in vivo data, and I already upload the slides in the website. Then later I will present them. Or
yeah, do you want us to present them now, or do you want us to present them later? So yes. The updates on the in vivo data. So we do get the larvae now. So we started screening and we have updates on the different compound series.
Okay, sure. Would you like to just share that with us now, Mark?
Yeah, of course. Let's share our screen. Okay, can you see our screen? Yes, okay. Yeah, today is my presentation of screening compounds from MycetOS Open Source in Galleria melanella larvae. Yeah. Yes, we got the compounds from MycetOS Open Source. Due to the in vivo data for MycetOS Open Source compounds, these compounds were selected as follows. 3 compounds from series 2, 3 compounds from series 5, and 4 compounds from Nature Products, and 3 compounds from MMV Global Health Priority Box. Then the detail will be showed below. Yeah, okay. At first, this compounds from series 2. We evaluated compounds from series 2. Then we got 5 compounds has a potential to inhibit fungal growth. But in the meantime, as for FS, just 3 compounds were selected, and these 3 compounds is also inhibit the fungal growth against the MM. So these 3 compounds were selected to do in vivo test. At first, we would test the toxicity of those compounds. Shown in the figure, these 3 compounds were tested at 20 micromolar. Yeah, there are no toxic. They are not toxic at 20 micromolar. And then at first,
the screen is frozen for some reason. Do you mind resetting the sharing option?
I think it's showing the wrong desktop. It's not the one you're presenting. It's the other one. Can you swap it over? Sorry, I didn't realize that as well.
Okay.
I think it's now the correct one.
Yeah, it's
now. It's the correct one.
We can see it now.
Okay. 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 So it's in vitro data before this summer. Then this 3 compounds were selected. Yeah. Then this figure is about the toxic test. These 3 compounds were not toxic at 20 micromolar. So at first, they will be treated in MM-infected larvae first. And then it's the compounds from series 5. 5 compounds were selected against MM, but just 3 compounds also has the potential to inhibit FS, and these 3 compounds were selected to do further test. Yeah, and of course the toxic test is the priority option. Yeah, and in this left figure, we can see these 3 compounds were tested with 2 different concentrations. 20 micromolar and 10 micromolar. Yeah, in this figure, you can see the 3 compounds at 20 micromolar. The infected, the treated larvae were died.
So they were toxic.
Yes, they were toxic. Yeah, and in the lower concentration, they has no significance compared to the aquarius. It's an untreated larvae. So the 10 micromolar were selected to do further treatment. Yeah, in the left figure is about survival curve. You can see the blue and the green one. These 2 compounds were prolonged. These 2 compounds prolong the infected larvae survival. Yes. And next it's about the natural product. 4 compounds were selected because this had the potential to inhibit fungi both against MM and FS. Of course, the toxic test is first be tested. It showed those 4 compounds were not toxic at 20 micromolar. So the treatment in infected larvae against infected larvae will be performed soon. Then is the MMV Global Health Priority books we got in 2023. Totally 240 compounds were included. In vitro test, we tested them in higher and lower concentrations first. Then 3 compounds were selected to test IC50s against MM and FS. 3 of 7 compounds IC50s were below 8 micromolar against MM, while all IC50s were above 20 micromolar against FS. So 3 compounds were selected to do further tests just in MM infected larvae. Yeah, this is in vitro data in figure. So these 3 compounds were tested in toxic test first. Just MMV 1 at 04559 was toxic at 20 micromolar. And then the concentration decrease in 10 micromolar. They are no toxic. So the further treatment experiments were performed, shown in the right figure. The MMV 1904559 did prolong larvae survival, and the value is in different significant difference. P is 0.03. Then it's about the detail of MMV of these compounds. In the meantime, considering itraconazole was the clinical use of mycetoma, so we considering that whether these compounds combined with itraconazole will be prolong larvae survival. So the combined therapy MMV and itraconazole was performed, but just one time we will get the further results. In the near future, in vivo test 3 compounds from series 2 and 4 compounds from natural products will be performed in MM infected larvae first. Then 3 compounds and 3 compounds from series 5 will be tested in FS infected larvae, and a combination therapy of the MMA compounds and the itraconazole will be tested in MMA infected larvae. And a small question about the compounds. We got compounds from different chemistries. Compounds will be dissolved in DMSO as usual, but where it should be saved below 20, below 80, or just keep them in temperature.
So that's a question to you all. We get so many compounds now. Where do you would like to save the compounds? If you ship compounds, please also write it down in the letter so that we know. Any solids we have stored in room temperature, but when they dissolve, we usually tend to store them at -20. Because our MMV boxes also should be stored at -20. So is that okay for you, for your compounds too, or do we need another practice for that?
Yes, from our experiences with cancer research or cancer experiments, we also use DMSO stock solutions of drug candidates. We also put them then, when they are dissolved in DMSO, we put them into a freezer at -20. That should be okay.
All compounds with PCL are stored in the freezer as well. But most of them are stable at room temperature, especially Keroxanis. I checked this.
Yeah.
Yes, when they are pure powder, they are also stable at 4-degree or at room temperature. But if you dissolve them, then better you freeze them and store them in the freezer.
I don't think there's going to be any additional benefits for going below 20 because DMSO freezes at about 19 degrees, so -20 should be well enough to actually freeze whatever's in there. I think just freezer should be good enough.
Okay, then we will do that. Yeah. We will do that as standard practice, but in case you send a compound or an oil or something else which differs from that standard practice, please let us know.
Yeah, okay.
It can only be one problem if you freeze them, that the DMSO turns solid, and then you need to use some time to get it liquid again after you put it out of the freezer again for your next experiments. That might be the only problem.
No, we are used to that. So we put them out well in the morning when we do an experiment.
Is condensation contamination going to be a problem, usually a problem for you, or is it okay for whatever you're doing testing with? Because sometimes when you take it out of the freezer, there's also going to be water plus DMSO that's going to be in there. So just from the moisture, that is something that I normally try to avoid. But it doesn't really matter. I think it should be okay for your situation.
Usually we make stock solutions which
average concentration.
Yeah, with a high concentration, and we dilute them further into. Well, we divide them so that we don't take out the same tube each time.
Okay, so that's all for my presentation. Do you have any question of my results?
No, I think. Any questions?
Yes.
Yeah.
So you plan to test the remaining compounds in this season against the larvae you mentioned, like Series 2 and natural product series compounds, or is it? But does it take to next season, next year then, until you're testing?
Yeah. Okay. I will try my best to finish all compounds before this summer, not next summer. This summer. Yeah.
Yeah. So the compounds which were depicted over here, they were planned for finalizing this larvae season.
Yes. All compounds I showed here, I will try my best to finish them this summer.
We do our best and we plan to do it so, but in case the larvae supplier again fails to deliver larvae, then we cannot that's why we can't promise. But for now, the larvae supply was steady the last few weeks. So,
yeah.
I have a quick question. In this testing round, none of the Series 6 catechozymes made it to the in vivo stage. Is that right?
no, they didn't. They were not susceptible enough to make it to the vivo rounds. A new student will start this Friday, the 1st of September, and she will pick up all the remaining screenings for in vitro. And hopefully by the start of next larvae season, we might be able to do in vivo screening for those compounds. But well, the first priority would be to train her on the in vitro test so that we can select the best compound for next season.
And the next season is when? Next year?
Yes, in April.
Mm-hmm. Okay.
Is there any more question, or shall I move on to the next start point? Oh, next start point is Matt and Martin. So I think we're just gonna leave that one there. That's just again on the shipping, on requesting compounds from Epicam. So the third one is actually on. I don't think this person is here today. May Cyprian. That's the username. Sorry, I forgot their name. But yes, it's on to the potential protein target for that crystal. That post I made about the crystal structures of isobound STIP51 enzymes from various organisms. So do we have any update on that one? Because I haven't been following that post actually.
No, I don't think there are updates for that.
Okay, we'll just leave that for now. Okay, so
Actually, do you want me to give a quick update on the phenothiazines? Because I think we actually may have a way forward.
Yep, sure. Sure, Anthony. Let's hear it.
So, let me just get this. This should be it. I think you're just seeing the screen here, right, of this. So there was a study that was done at NEU, Lori Ferens, who Dr. Todd knows well. They were doing testing against amoeba, and they made a chlorpromazine analog, which is this one here, and it was potent against the amoeba. And this is not exactly a phenothiazine, but it's very close in structure to chlorpromazine. So Bernard, maybe you could reach out because Dr. Todd's on vacation. Lori said she would make a list of available compounds in this. So I guess we just need to reach out. And then I think one of the lecturers at a university said they might make 2 or 3 phenothiazines. Maybe this one here, the N-phenethyl. So it will be interesting to see, but these are a very unusual class of compounds. But if we can get samples, they might be interesting. That's about it so far. Just have to see if Bernard or Matt or someone can reach out because they have other functionalized things here too, not just tricyclics. They have these kinase type stuff too, which could be interesting. So they have all these different compounds, including the chlorpromazine analogs. They also have these kinase inhibitor type compounds too. So this is a very big library, so we can see if Lori could share it with us.
Okay. Yeah, I see. Yeah. Thanks, Anthony. I think we'll just note that down. Might need also Matt for further discussion. So because I'm not quite up to date with that, but yeah, let's just note that down for now. Thanks, Anthony.
I did share I shared the web browser, right? It was the right, yeah. Okay, but yeah, so that could be interesting to work on, perhaps. I just haven't heard back from Lori, and Dr. Todd is on a holiday, so You know, maybe Bernard, you could reach out to Lori, see what happened.
Yeah, sure, I can try. Yeah, we'll see. Maybe they're also on vacation at the moment, but I try to reach them.
Okay, sounds good.
All right.
I
Next one.
think in terms of kinase inhibitors, Wendy has received a compound library recently about kinase inhibitors because I read something on GitHub about it.
Yeah, it's also on the agenda. It's under any other business, but we can give an update now, or Hang, do you would like to stick to the agenda for now?
Yeah, you can do that. We'll have one fewer things to talk about later.
Yeah,
okay.
just a quick thing. So that library that was tested against the amoeba was a mix of different things. It had some tricyclic chlorpromazine analogs. It had kinase inhibitors. It was a mix of everything. It was like a MMV box almost. So yeah, if we then more narrow targeted kinase library might be a bit more. But if Lori can send over some compounds, especially the phenothiazines, because it doesn't seem like there's really any time for people to synthesize them, and they're quite expensive, despite being very simple, which is not great. But hopefully that will solve some of the issues, and we can see what happens.
Yeah, okay, that sounds good.
Mickey, could you give an update on the kinase library?
Yes, I can. So I've did 2 screenings. When I received the kinase library and I looked into their information sheets, they mentioned that the library is quite potent. So they recommend screening at quite a low concentration. So I did that first at 1 micromolar, and there was a few compounds which were like seemingly inhibiting fungal growth, but they didn't really come out in the eventual assay itself when we actually performed the measurements. So I also did a second screening with 2 micromolars, and there I had 1 compound which is relatively consistent between the Duplo. So I've started looking into that one this morning, and then there are a few which are inhibiting fungal growth, relatively okay in one plate and not really in the other. So I'm going to repeat the assay also at a higher concentration of 10 micromolars, but that's planned for, I believe, in 2 weeks or something. So I guess by next meeting we can give a bit of a clearer overview, but it doesn't seem to be super potent, at least within this library. But I guess that for now is most of it. Yeah, most of the updates that I can give on the kinase library.
All right, thanks, Mickey. Is there any question for Mickey? All right, so let's move on to the agenda dot points for regarding paper. So I should already talk about a few points of this. So Excel sheet. I actually I looked through the issue for this month's meeting. Is the link to the Excel sheet in a different place? Because I don't actually see it here. When did you where did you post the link for Excel, the spreadsheet?
I put it on the manuscript writing issue.
Okay.
I will put it in the Dropbox as well, and then link it here.
Oh, okay. Yeah. Thanks. Dimitri, you had a chance to upload your lab notebook to the UCL repository, or is that still?
As I said before, it's within Professor Todd jurisdiction, because I don't have the access to that option.
Okay,
It's a recurring issue, so it's like unless he resolves this, there's no way I can upload it on my own. But he knows that, so that's why it says here like that Matt will assist like with transferring data.
Okay, so you guys are still kind of fixing that. We'll just leave it there.
On my side, like everything has been updated on lab archives, so he just needs to put the file. That's it.
Okay. Excel sheet, that's good. Final update data to the master table. Is that already
Yes,
done?
I did that this afternoon.
Okay.
She sent it already to me. I will upload it this afternoon.
Okay, so myself and Dimitri will check the table. I haven't checked it yet. I will do that when I get to that section. Oh yeah, I think that's most of the stuff done on the paper. So next one is on the siderophores, which is because a core contributor to report back on potential structures in the original issues. Do we have any updates on that?
Yeah, we received a fluorescent siderophore from Professor Doyle, and we're going to screen it in the near future, in vitro and hopefully also in vivo. But yeah, that's ongoing.
Okay, thanks. And the second point, which is so that's.
Are you able to do in larval fluorescence if they are cellular, so you can track it, or
yes,
would that be?
we could do imaging on the larvae directly, or we can simply make histology slides and check the fluorescence then. So the histology, if you take a frozen section.
So next one is going to be on series 2. Clementine is not here today. Anita, do you actually have any more updates on this? Because usually this one goes to Clementine, which is not here. Just double checking.
I asked her, but she didn't have anything else that she added. Just the fact that she updated the wiki.
Oh, okay. Sounds good. So Matt to organize meetings. Let us back to Matt. Andy Bishop and Julia are not here today, so we can't really ask them for that. Or how many samples are likely to be incoming. Okay, so some of you just have to just reassign them to Clementine and other people. So I don't think we can do much more on this one, unless anyone else has anything they want to add in terms of things for series 2. Otherwise, we'll just move on to series 5.
Series 2 aminothiazoles from the MSc students who worked as part of UCL master's project have been sent to Ma and Wendy to Rotterdam with the latest batch of caroxymes. So it was like a large shipment. And the list is updated, so the unique MycetOS codes, everything. And same applies to the Vidgert compounds. So we selected one of the best, the purest samples, and then it was sent along with the shipment.
Okay.
Essentially, everything that was planned to be shipped from UCL has been shipped by now.
Okay. Okay, thank you.
I think Seven Oaks will be working on new synthesis when the new term starts. So with some new students, I guess. I think they mentioned this to Matt some while ago. But we will double check with them in the near future before the next meeting.
Okay, sounds good. Yeah, there's a few action items assigned to Dr. Cruz on obtaining samples, so both in series 2 and series 5. Is anyone actually talking to Dr. Cruz on these action items?
I was in a CC on the compounds which were active against Candida. So she is looking around for compounds for us to screen. I'm not sure exactly which set of compounds they were, but yeah, there's some activity going on there.
Okay, so but it's ongoing.
Yes.
Okay, sounds good. Okay, so this one's Marita to search for additional samples could be obtained from other labs. I haven't actually heard personally from Marita on this. I might have to chase up Marita. Anita, do you know about this particular action items on Marita?
So she did post a few hours ago, and she basically just said that she's still looking into finding other groups that have made the compound.
Okay. Yeah, I might have to. Is that on a separate GitHub issue? Or is it down in the comment section? Actually, sorry, I haven't had a look yet.
It's like at the bottom.
Okay,
yeah.
I see.
She's included her slides as well.
Okay, awesome. Okay, so Marita is still looking into that. We'll just leave that one there. Okay, next one is Andrea to clarify the new set of benzoxazole that has been shipped to Wendy. Okay, so Andrea is not here today, I think from the memo, so we'll just leave that one. Next one is asking for Payan to actually join GitHub. I actually haven't heard back from Alice yet. Alice actually told us last time that I think Alice will update us when Payan is ready. I might chase up Alice or Payan. Alice is also on medical leave right now, so she won't be like actively on correspondence. But I'll see if I can, whenever I see her, I'll double check. But I think last time I heard Payan was not quite ready yet, and I don't have an update on that actually. Okay, last one is on Marita again, so I think we'll just leave that there. There's 10 minutes left. Let's go for series 6. Is there any update on series 6 on GitHub Science?
Just the fact that I sent the latest batch of the finalized compounds to Wendy. So it would be the third batch for series 6 GitHub Science. We'll be just waiting for the potency results from now on onwards. There are a few more methodology tests that are being carried out before I finish my PhD. So that's on the stability of the isomers. And coming back, a few words from the question that Wendy asked about the stability of the compounds. So for example, catechozymes, at least one of the ENZ forms of one sample have been showed stability up to 120 degrees when heated in DMSO. But this is ongoing, so probably share some more results next time. But definitely stable at room temperature and after storage in the sub-level, sub-zero conditions. That's for sure.
Okay, thanks, Dimitri. And there's one miscellaneous on DMPK. Do we have any more updates on that progress? So last time I think it was on shipping compounds to selecting and shipping compounds. Do we have an update for progress on that as well?
Yes, everything has been shipped as well, mainly from UCL, I guess. Just showing you that quickly. So that's the this is one of the that's one of the batches. That's the second one from the reserve. So we have essentially series 1 phenaremols and catechozymes as well. The second batch was supposed to be supposed to have additional compounds from University of Sydney, but Hank, as far as I understood, you probably did not have them, or could not get them on time to download.
Yeah, so I only have one compound that's also already a repeat of was in the discussion. So in the end, I was like, yeah, that's not it's not feasible just to ship one. And the other two I ran out, so I don't have enough. So yeah, in the end, yeah, I won't be able to ship any.
But the good point is, like, for at least for series 1 phenaramols, most of the compounds that will be tested for DMPK will go into the manuscript. So that's like additional, very useful set of information that we'll get. And luckily, I tested them before sending them to Dundee. I retested all the compounds for stability and can confirm now that like most of them after 4 years of storage are absolutely stable, and that has been confirmed by NMR, which is good news. So their shelf life is quite long, at least for now.
Yeah, definitely. The green one in the middle, I also have a lot of that one. Yeah, and they just stay there forever. I don't really do anything. Actually,
one of the positive controls. It's not everything that I did personally. We have like a few positive controls, and MycetOS 10 is one of the most potent compounds in the series as well. So that's sorted. For DMPK, we'll be just waiting for Dr. Lauren Webster to come back with the results, and she said she will post something as soon as they start.
Okay, yeah, thanks. Is there anyone else who would like to add to this or any other business? I think that's really the last section on the agenda. All right, sounds good. So I think that's all from us today. We are 6 minutes ahead of time.
Bye, everyone.
Okay, bye, Anthony.
See
Is
you
there
later.
anyone else who wants to share anything else? Otherwise, we'll adjourn this meeting. Who is sharing / writing? Sorry, taking minutes for next time. Do we have a roster?
Yes, we do have a roster. It's displayed in the GitHub page on organizing meetings.
So I think it should be UCL that's sharing next time, and
Us taking minutes, I guess.
okay, sure. All right. Thank you very much. Okay, so if there's nothing else, then I guess we're done for today. Thank you very much for everyone for joining. Lovely to see everyone as well.
Bye, everyone.
Wendy, can I have a quick word with you after the meeting, just for 1 minute, if
Yes.
you don't mind? Thank you so much. Goodbye, everyone. Thank you so much.
All right,
See you.
goodbye.
Bye.
Just a few words about the compound, the mixed isomers that we had before this